Right, good afternoon. I hate giving talks just before lunch or after lunch, but actually I have no choice right now. So I'll try and keep everyone interested and try to finish just before lunch, actually. So I've been working on blood parasites for the last 10 years or so. And I'm actually a trained ecologist, but I got interested into blood parasites when I actually embarked on a postdoc, which was 10, 12 years back. And all I want to do is to learn genetics and to understand how the DNA and everything works. But when I started getting into these parasites, I liked the system so much that I'm still finding more questions and trying to work out what all these parasites are about. So I'm mainly interested into, so right now I'm structured this talk into, which is based on 2 studies which I've done in the last few years. I mainly was interested into looking into the parasites and how the host colonization and parasites work, because there have been a lot of studies which people have done, and they talk about parasite invasion and host invasion. But with respect to blood parasites, I don't think it has been looked into in great detail. And I'll tell you why it is important and why I think it was a really important question to look at. So when you look into biological invasions, I mean, there are a lot of species you can see. Pointer. Like this lantana, which is very common over here. It's one of the invasive species. So wherever you have these introduced species, they usually have larger population densities. They tend to acquire bigger body sizes. And they also compete with the native species, and that's how they progress. And also, one of the reasons have been given for their successful invasion is their escape from the parasites. And when you look into an island or any other situation, the success of most of these introduced species has been given as because they have lost pretty much all of their parasites. In fact, when you look into these species in their native range, they have more parasites than what you find in the introduced populations. So how does this whole species introduction and the parasite introduction work? So look into a scenario. Say if you want to introduce, say, some species of a bird to a new location, what are the chances of it taking its parasite with it? So the chances are that you release a few number of birds there, and then they probably are not infected. So obviously there will not be any transmission of parasites to new locations. The other scenario could be that you release the birds, but there have been severe founder effects. So there was loss of population. So whatever parasitemia they had was lost. So there will not be any parasite transmission. But from parasite perspective, it's a parasite which can be vertically transmitted, just like from mother to offspring or directly transmitted parasite. It should be fairly easy, right? I mean, as long as the parasite is not very host specific. So it can be transmitted from one host to the other host. But there's a real problem for these horizontally or, sorry, it's the other way around, for parasites, where if the parasite is dependent on a intermediate host to complete its life cycle, and that's the parasite, the vector-mediated parasites, which I am actually interested in. So there are 2 main groups of parasites which I would like to talk about today. Is the the species of Plasmodium and Hemoproteus, and they both are very widely distributed and cosmopolitan in distribution, and they infect a large number of bird species around the globe. You just name it, they are there everywhere on this planet except for Antarctica, for obvious reasons, because the vector species, the mosquitoes or midges, whatever they rely on their successful transmissions, those vector species are not found there. And they also share a very close evolutionary history. But before I go into that, so look at the slide on the back of the backdrop of this one. These are the cells of a bird species infected with these parasites, and all of these purple dots are Plasmodium parasites, which the bird has been infected with. And this is how traditionally people have been studying parasites, that you go and you make a blood slide, and then you look into under the microscope and identify those parasites. And based on that technology or technique, they realized they said there were only around, say, about 175 species of parasites so far known for the bird. But in the last 10 years, when they embarked on this DNA technique and they managed to design their primers and screen these birds, they realized the diversity is much more than what they thought. So at the moment, they think there might be more parasite species than what species of birds you have actually on the planet, basically. So that's the amount of diversity I'm talking about. So there are like n number of species of these parasites which infect birds, and we have no idea where to start with and begin to look into that. And the reason, so they shared a close evolutionary history in, say, so you can see, looking at this tree, that the top, for example, here, the red branch is a Plasmodium falciparum, which is a parasite which infects humans or also the non on the primate. And the diversification of this parasite, because it relies on these Anopheles mosquitoes or Anopheles mosquitoes. And then you have this whole blue, the bird malaria, which relies on these culisine mosquitoes. So this is the most point of diversification between culisine and Anopheles mosquito, because these two parasites rely on these particular vector species. So what I'm trying to say, that Anopheles mosquito cannot transmit a bird malaria, and a Anopheles mosquito cannot transmit a bird malaria, or culisine mosquito cannot transmit a human malaria, basically. That was the point of diversification for these parasites. And then you have these parahemoproteus, hemoproteus, which rely on these midges and sandflies and so on. So you have all sorts of malaria, like primate malaria, rodent, bat, lizard, and bird, all across the globe, which relies on a specific vector species. And when a bird is infected with these parasites, either Plasmodium or hemoproteus, then they do have certain physiological effect. They do have anemia, fever. If it's a acute infection, it does induce a immune response. It does affect their reproductive success, and in a very, very extreme cases, it does cause death in this population if the birds are severely infected. And I can't ignore this particular case of Hawaii Islands, which is actually synonymous with avian malaria, where just because the island is so isolated from the mainland, and the bird species have evolved in the absence of all the blood pathogens, and with the introduction of all the new species which brought malaria with them, and also the introduction of the vector species of mosquitoes, especially the one species of mosquito, which has led to the mass mortality in these native species there, which has led to the extinction of the many 50 to 95% of the endemic species have died. So that is the classic case of where biological invasion can cause extinction of species. And so I've been working on originally on this, where and how this malaria got introduced into Hawaii and where it came from. But while going through all of that records, I also stumbled upon a very interesting scenario of these enemy release hypotheses, especially with respect to this particular species of bird, which is a common Myna, which is very common. I mean, if you go out right now, you see hundreds of flocks of this bird. The interesting thing about common Myna, which when I was looking at its record, that they have introduced this species into several parts of the world. And from the parasite point of view, I mean, again, going back to the same thing, that the success of a species, as they say, relies on its release from its enemies and co-predators, and also parasites. So basically, they release this common Myna into these different countries, from India, which is the native place for this bird, all the way to Hawaii, then South Africa, Australia, New Zealand, Fiji, and Cook Islands, as a control for insects, actually, and also to increase diversity. We know like a lot of species of birds have been introduced in New Zealand right now just to increase the diversity, because they thought there was not enough birds there or some species there. And this has been done very well documented over a period of the last 200 years. You can see all the dates are very well documented. And in that process, I thought the birds might have carried parasites with them from India at all, if those introductions were done in a sequential manner. I mean, say they released 100 birds. If they were not got established there, they probably came back to the native site. They got 30 more birds. So it all happened in a series of events over the course of certain periods. So like, for example, in Hawaii, when I was reading the account, that it took them 10 years to make sure the Myna was properly established population was there, basically. So I don't know how many sequential introductions were done in different populations. So I went around and I had enough data for all these parasite species and from all these locations, because I was looking into the parasites for the Hawaii where they came from. And I started screening the DNA and using this mitochondrial gene, which is a very variable region which we have found in these parasites for both for Plasmodium and Hemoproteus. You can get the DNA, you can sequence it, and you can look into the sequences where it will fall in the whole from the phylogenetic perspective. So I went and sampled other sites within India, and these are the main sites roughly where the birds have been introduced from India. So here you can see from Assam, Delhi, Haryana, Maharashtra, so all those populations we had. And this is just the basic prevalence data which I was looking within India. And on top of you can see the number of samples I have, which I collected over the things. So within India, I didn't find any difference in the Plasmodium or Hemoproteus prevalence, basically. So, which is fine. I mean, I was not expecting anything here. It's just a snapshot in time that there was no prevalence in this. But if I compare this data across the region also, then also I don't see much difference actually across the region. But if you just compare across India with rest of the population with the introduced region, the only difference you find is with Fiji or maybe Hawaii, even though samples are the same, but there is only one lineage of parasite there. Or say with Cook Island with a little bit there. But the interesting thing for me to look was to where do these parasite lineages actually fall with respect to each other? Do they show any geographical structuring here? And just let me talk you through this tree. So here, these are all my Myna population: India, Australia, New Zealand, South Africa, Cook Island, Hawaii. And I try to compare them with the parasite lineages I found in other host species, which is the non-Myna species, so to say here. So for example, this South African lineage I found in 2 individuals here in South Africa, and these ones here, and so on. So just by looking at this tree, the first thing which comes to mind, you'll see that there is a very large parasite diversity in India as compared to any other introduced region here, which actually proves your sort of the enemy release hypothesis that in native region the diversity is much higher than in the introduced region. And the second thing I found very interesting about this lineage IND9, which is actually shared between India and most of the other introduced region here, including the Hawaii here, because Hawaii has only one parasite lineage so far which has been found, has been the main culprit for extinction of those birds on that island. And that actually is a sort of an indication that Myna has carried that lineage in those introduced regions, as opposed to here, this IND2 lineages. But then there are also singleton lineages as well, which I have found, like the one over here in New Zealand, or say this one here, just on its own in Australia. These are probably the lineages which Myna, they've got infected with the introduced when they were introduced in that region. But the other interesting thing about this tree is that there is not a single parasite lineage which I have found in the introduced region in Myna, but not in the native region. So that actually tells you that most of the lineages which Myna have shared across, they are the ones which they have carried with them to the introduced area. And a lot of the singletons, they have got infected over there. But so basically, in terms of looking, the source of the infection is actually indicating all the way back to India over and over again. And the other way to look at it was, if you think that India has been the source of these parasite lineages, then the genetic divergence or the lineage this should not be higher than, they should be more genetically similar. So here you see that the mean divergence across these populations is much lower or pretty much similar to India, and that sort of signifies that India probably was the source for the transmission of these parasite lineages. So this is just to summarize that obviously there was some evidence, and India has the high parasite diversity, but I can't differentiate because these parasites are so non-host specific. I mean, I can't conclusively say that these parasites which were there and they got introduced were not for India, because my sampling for India is not so thorough, basically. Okay, so this is not where I'm ending my talk. This is just to just give you some perspective of that. Now, while doing that study, which I did, I mean, it really made me think that when we look into the literature, a lot of the work which people have done they always talk about these introduced species, which have been human-mediated introductions, right? Somebody released X, Y, or Z species to new areas, and then they looked into these parasite species or any sort of basically biological phenomenon there. It really made me think that what happens in a natural range expansion when if some species is trying to expand its range from A to B area, what happens? Do they also carry their parasites, and what sort of diversification or what of new parasitemia they encounter? And considering this whole blood parasite scenario here, which requires a intermediate host, or actually I should not call it an intermediate host, it's a proper host, because without mosquitoes they can't transmit to the next host bird species. And if the parasite is non-host specific, like these blood parasites, what sort of pattern I'm going to get? So here I actually started looking into these parasites in the southern Pacific islands. So this is Australia here, and here is you have New Caledonia and this Vanuatu island. Now, I just didn't think one night I have to go and work on these islands. There was a reason I decided to select these places to work. So there's a species that's called Silvereye, Zosterops lateralis. This is how it looks like. It's a very common species closely related to it, Zosterops palpebrosus, which is found very common in India right now. It's the commonest bird you can see outside as well. So Zosterops lateralis is very common in Tasmania, and it has colonized from Tasmania to Chatham Island. You can see the dates. People have documented all of these colonizations from here to here, then to the North Island, then to the Chatham Islands, and then here. And they've also looked into their host genetic diversity, what sort of alleles loss or patterns they have faced during this colonization. But then the same species from Australia, which is this distance from Australia to New Caledonia or to Vanuatu is roughly 1,000 kilometers. The species more than 1 million or 2 million years ago has colonized these islands as well, okay? So I wanted to look into, do they have carried parasites from here to there? Because it's a natural range expansion. Nobody has taken them or released them there, basically. So I have samples from over like around 1,000, from 23 localities, like 1,000 samples. And so here you have Vanuatu Island. So just put things as it is Z. lateralis, which is found in all of the islands in Vanuatu, and also in mainland Caledonia. And then you have Zosterops flavifrons, all in greens. So you can see some of the islands, they do share this sympatry with this lateralis there, except for, I think, one place where they have gone, from here, from Anatum Island, they are not there. But they used to be there. We don't know why. They just are not there anymore. And also Mavo Island on the top. But flavifloron is endemic to Vanuatu Island. And then you have a third species, which is endemic to New Caledonia, which is Zosterops xanthocroas, which is found in mainland New Caledonia and also one of the islands of New Caledonia, which is the Mare Island. And then you have two more species, which are endemic only to these Lifu Islands, basically there. So do you have any questions? You seem to be deep to discussion. So Enornatus and Minutus are only found in Lifu Island. So you can see it's a perfect situation to explore this natural range expansion into parasites. And what is I find very exciting and unique about this whole system here with respect to the location of the islands and the species, in an island system you can tease apart these processes very well as opposed to in the mainland, because in mainland you have all sorts of things going on, but these islands are far isolated. They are distinct entities, so to say, so you can see these problems. And also, not to mention, they also have their own endemic mosquito species, so which I'll come to that in a bit. So after screening the samples, whatever I have for these bird species for the parasites, this is what I got. So you can see I've arranged them. So this is Australia, Tasmania, New Zealand, Chatham Island, Norfolk, Vanuatu, and New Caledonia. All the yellow corresponds to the species which are parasite lineages which I found in lateralis. All the blue corresponds to xanthocroas, and so on. And the green ones, you can see how few. I'll come back to that. And also, this is by the lineages. When you have a phylogenetic tree, So I said I'm an ecologist. I just won't rely on this phylogenetic tree, right, to see, okay, this is what the species lineages I have found. I also want to know whether I have sampled enough to get all the lineages across the species. So I've done this rarefaction or species accumulation curve to look into the different species. So you can see for lateralis, I am quite far below in sampling all the lineages what I could have found. So 23 is the minimum what the results gives me, and as opposed to Xanthochrous also I am so below. It's like it's for 10.22 and I have only 8 things. But from lateralis from across the Vanuatu island, if I look into it, except for New Caledonia, where I am far behind, I managed to sample pretty much all the lineages what I could have. So that gives me a confidence that I have pretty much, for most of the sites, data which is good enough to compare across these species. And from the regional perspective, from the mainland Australia, you can see I've got only 3 lineages, but it is within the species point of view, this is good enough. And also from Tasmania, except for, again, in Vanuatu, I'm close, but not in New Caledonia. I was far behind in screening all the lineages. Now, going back to the sharing of these parasites, that's the question which I was trying to understand, whether the birds which colonized from Australia to Vanuatu and New Caledonia, which was estimated to be 1 million, 2 million years ago, have they carried any parasite with them? So you can see that only this Australian lineage which I found, there are 2 lineages of Plasmodium I have, and there is 1 Haemoproteus lineage which I have found here, which is 36 Zosterops species have got this lineages, and only 4 of the Vanuatu I have found, and none of the other, and including of those 40 Vanuatu which I found, 4 of those flavifrons which are endemic to Vanuatu, which means there is Zosterops lateralis which colonized these islands. They probably carried these lineages with them. And just below that lineage, which is V and 1H, which is barely 1 base pair different to this one, which is so prevalent and common across these islands, basically. So, looking at the mitochondrial and the nuclear DNA concordance between these lineages, we know that these are potentially biologically independent units. So that's why I'm not calling them species. I'm continuously using the word lineage here. But they are potentially species. We need more data to verify them up to the species level, basically. And what I am actually showing you in terms of this parasite lineages, we still are beginning to understand how these all parasite diversity works across these birds. So I would say this is just tip of the iceberg what I'm trying to understand here. And there are also some unique lineages I found, say for Mare Island, only 1 in New Caledonia, and like these 2 lineages only found over there. Now, for the recently colonized group, if you look at it, there are a lot of species which colonize the Zosterops which colonized from Tasmania all the way to Norfolk or Chatham Islands. They have very good, perfect sharing with the species of the parasite lineages. And over here as well. But also Chatham Island has its own parasite lineages as well. So again, it comes to you back to the point that a bit like in the human-mediated introduction, these natural range extensions did manage to get some of those lineages across to the new places. But however, this less number of lineage sharing between the mainland Australia to the Vanuatu and New Caledonia, it could be one of the reason for that could be that because these colonizations probably have not happened as many times from the mainland as these birds have been colonizing from Tasmania. So when you have continuous host introduction going on, so these have been, based on the literature, they say these are the larger flocks which have been colonizing these islands. So this is the host genetic data they have looked into from Tasmania. You can see that there was hardly any allele loss, which means the large founder population were going there. So when there is no bottleneck in the host population, the loss of parasite would be very less, as opposed to if you introduce the bird populations go there, if there be bottleneck, so chances of parasite is lost. So this just basically indicates that this sort of fits with my data that because these larger population of hosts are continuously supplementing the island population, those parasite loss was not seen in these populations as opposed to from Australia to Vanuatu. But the one way I also want to highlight this thing here is a lot of studies, when they look into these parasites and transmission, they just go and catch birds, and then they analyze and they present the data, which I feel when is a parasite system relies on, oh gosh, when it relies on two host species, basically, which is mosquitoes as one of the main host species for these parasites. Without this, they cannot complete their life cycle. In fact, most of the reproductive isolation of these parasites is inside the mosquito, not inside the bird host, basically. So which means these parasites can't reproduce if you don't have these mosquito species. So clearly, this sort of makes you think that some of the lineages which have been successfully transmitted, you have to have that competent vector to be there for the successful transmission to these areas. So to complete that jigsaw puzzle, I actually went and did some mosquito sampling around some of these islands in Vanuatu and in New Caledonia as well, and trying to piece apart, will I find the same lineages of parasites in these mosquitoes or not? And just to give you a brief summary of that, I don't expect you to read these names of mosquitoes, but just roughly tells you the infection rate of parasites I've found in these. So here you have 14 different species of mosquitoes, and then you have all the 4 Zosterops, which I have found the parasite lineages. So for example, this particular lineage has been common across so many mosquito species, and it has also been found in Zosterops, and it also tells you that it can be transmitted by multiple vector species. That's why it is so common across these Zosterops species, as opposed to this particular one, which is found in this particular mosquito species called Aedes notoscriptus, which is actually an endemic mosquito in New Caledonia. Hence, I have not found this particular lineage in any other host species, so I think it is probably endemic in New Caledonia as well. And some of these species, lineages I found only in some of the Zosterops, but nowhere else in the planet. So, and also, I mean, you might argue also with me that, so this is just a small data set I have from these islands. So how do I know that these species are endemic to these areas, not anywhere else? I mean, by now, in the last 12 years, there have been so many studies on these parasites have been done all across the globe that this data set we have is so, it's like over 1,500 or 1,600 or 2,000 lineages of parasites we know right now exist there. And when you compare across those lineages, you can tell where these parasites are lineages or what host species they can be found in. So to conclude this bit, so Zosterops populations which established which is the recent colonization, they do not show any reduction in parasite because they show a lot of identical lineages or closely clustered lineages with their source and colonized population. However, with respect to the anciently colonized Zosterops species where I found in the elevated levels of parasite diversity compared to the mainland. So the species which colonized probably is got more infected with parasites in its introduced range or its colonized range than it was in Australia, which was in contrast with the introduced population which I have seen. And this is because these archipelagos have been colonized several times from the other bird species as well. So I think that's why they are so rich in parasite species as compared to the other areas. And that's it. I'll take some questions if you have.